All right. What did we get? Most members back. So,
Hey, how you doing?
so we have Dr. Eric Roy here and Kate Porterfield here with us in the room. And we invited them to talk, just give a general overview of their microplastics research in Vermont, and to give you, Eric and Kate, I gave you a little bit when I asked you to come, but basically the AIB has just spent the last year and a half really doing a deep dive into neonic treated seeds, which was asked of us by the legislature. It's depending on how things go in the legislature in the next few months, we might have another ask for neonic information to be able to give a recommendation for that for more best management practices. But other than that, we're looking for our next topic to address. We did last Tuesday, there was, through the Pesticide Safety and Education Program at UVM, we had an agricultural applicator survey training, and so we did like a poll during that virtual training asking what concerns you, basically. And microplastics was kind of like number 3 there. Number 1 was non-target pollinator exposure, as far as majority of answers, but microplastics is top 3. So
What was the other one?
yeah, sorry, I just closed it too. PFAS was just below microplastics.
The disposal of agricultural inputs, so like tires, pesticides, bale wrap, other kinds of things associated with the industry.
Yep. Other choices were non-target beneficial exposure, so not necessarily pollinator, but other beneficial insects. Non-target rodenticide exposure. PFAS, microplastics. I think those were the choices. On a Zoom poll, we couldn't, they don't have free text, so but only 2 people picked other out of 55. So that's decent. Yeah. So without further ado, I'll hand it over to you, but maybe give a quick introduction.
Yeah, so for everybody online, before I share the slides, I'm Eric Roy, Kate Porterfield behind me, and we are going to provide kind of a pretty broad overview, some details on some of our studies that we've conducted at UVM on microplastics. My guess is we'll have plenty of time for questions and discussion afterwards. So I will... Okay, so we'll get right into it. Kate, what year did we start doing this work?
Maybe 3 years ago at this point.
Yeah, it feels even longer than that. It's been a journey. So we'll just give you some of the high points. So first, I just want to summarize the different aspects of our research at UVM on microplastics. So when we got into this, it was new for us and new for a lot of folks. And the first step when trying to kind of enter a new field is to see what's out there in terms of the literature. So Kate, myself, and others dove in and did a comprehensive literature review on this topic. I guess just to take a step back, the reason we were so interested in this is because we were seeing it come up over and over again, and some of our work focused on food waste management. Our lab has primarily over the years focused on beneficial ways to capture resources and recycle nutrients, also generate other beneficial products like biogas or compost. But increasingly, there was concerns about plastic contamination, new technologies coming online that were a focus of some of those concerns. And so we decided if nobody else is doing it, we should. So we dove in first with this literature review, reviewed approximately 150 papers in total. We are really focused on, either products or intermediate products of food waste recycling and agricultural soil. So microplastics has been pretty well studied, although there's still a lot to learn and a lot going on in water. For example, the science is just further along when it comes to microplastic contamination in water. There's steps being made in soils, but we were finding that this particular area of food waste derived products like compost or digestate, there was just not as much out there, and we were trying to gather everything we could. And so in that literature review, we're really focused on collecting information, interpreting that information, and then making some recommendations for science and policy. And we'll share a little bit from that soon. The next thing we did is dove in with our own primary research, so to actually quantify microplastics in different materials. So that included depackaged food waste, digestate, and compost. And this was achieved across a few different projects. One of those projects was really focused on mechanically depackaged food waste, thinking about both the biogas potential of that material in anaerobic digesters, as well as the potential microplastic contamination. And then a subsequent study focused instead on compost. So we call this a statewide survey. It included 20 composts from across the state, all from commercial facilities where we quantified microplastic content. And then the third project listed here is currently underway. It's an EPA-funded project that includes more research on the biogas potential of different food waste and beverage waste streams, as well as microplastic characterization. And then the most recent work we've started and are making good progress on is there's been a long history of trying to evaluate different food waste management methods with life cycle assessment, so composting, anaerobic digestion, incineration. For many years, people have tried to understand what are the pros and cons of each approach, how can each approach potentially be enhanced or made more environmentally beneficial. And so we've dove into that work, but with a unique angle where we're including kind of plastic flows within that life cycle assessment. So plastic contamination is now one of the multiple environmental burdens that we're considering when we think about the life cycle of different food waste management strategies. And so just to say a little bit more about the review paper, this is open source, available online. If anybody needs me to send them a PDF, I'm happy to do so. Published a couple of years ago in the Journal of Environmental Quality. Included contributions from Kate and I, as well as Sarah Hobson, Deb Neer, and Meredith Niles at UVM. And so just to summarize some of the high points from that review, so we were only able to identify 16 studies, and this is not just limited to the US, this was anywhere in the world, that provided original data on microplastics in organic residuals. And so by organic residuals, I mean food waste, compost, digestate, focusing on compost and digestate derived from food waste. So we're not necessarily focused on materials that were derived from wastewater biosolids, which is kind of, we're treating this kind of a separate issue here. And when we looked at the data that were available across those studies, those that provided data in terms of counts, so counts are particles per dry kilogram of material. So that's an individual particle of plastic. How many of those are in a dry kilogram for these different types of organic residuals? And the first thing we notice is just a really huge range for many of these materials, several orders of magnitude difference between the low end and the high end, and that they were overlapping. All of these ranges for these different materials were overlapping. So we weren't necessarily seeing in the literature clear evidence that one type of organic residual tends to be more contaminated than another. When we look at this by mass, which I'll say a bit more as we go, but I think this is the best way to look at microplastic contamination, because you can have a small number of particles that are relatively heavy, and that adds up to a lot of plastic, or you can have a lot of little teeny particles that really don't add up to much weight. So because any larger particles we add to the environment will eventually become smaller particles themselves, to me what's most important is what is the total mass of plastic we're adding to the environment. So I like the percent by mass numbers. And those also varied quite a bit in terms of what was available in the literature when we conducted this review, from really, really small numbers up to, 0.14% for compost or 0.25% for digestate. And then food waste varied quite a bit, with some of the highest numbers being from some estimates for source-separated household bio-waste. And, one of the key takeaways that we came away from this review with, which we shared with Agency of Ag and others in the state, is that this really appears to be a systemic challenge, not limited to any one food waste processing strategy. So, most of the places people were looking for microplastics, they were finding some, and it varied widely how much they were finding, but the contamination was pretty widespread wherever people were looking. And some additional key takeaways: variabilities in estimates can likely be driven by multiple factors, including the feedstock to the composting or anaerobic digestion operation, the processing, both in terms of how exactly packaging separation is occurring, whether that's human labor or mechanical depackaging, as well as other processing methods such as screening that might occur for some of these different technologies. Those can influence what ends up in the final products. And then very importantly, the methods used for quantifying microplastics can lead to the variability as well. For example, some studies could go much lower in terms of what size microplastics they were able to observe. And if you can go lower, you will find more. So those studies would tend to report, higher particle counts, for example, than studies that focused on larger size fractions. No standard methods, and this is still true to this day. It's been a few years. We've made progress. I think others have made progress, but it's a little bit wild, wild west. There's researchers using a variety of methods for isolating, identifying, and characterizing microplastics in complex organic matrices. I won't get too into the weeds on this, but the key words in that line are complex organic matrices. These are really high organic matter materials, and that creates some significant challenges when it comes to isolating microplastics that are not necessarily present when we're looking at water or mineral soil. And units matter, and we'll show you some examples of why this is the case as we get into our primary research. There's just kind of this disconnect where we have some researchers only focusing on abundance, so only focusing on counts, others only focusing on mass. Only a third of the studies we reviewed at the time looked at both. But the policymakers tend to be focused on weight-based limits, and again, I agree with that. I think that's the right focus. But it means there's a chunk of literature out there that doesn't really help us much because it only provides count-based estimates. So when we're trying to figure out where do we set that weight-based limit, we have even less information to go on because some of the studies are not providing the information in that format. And then we did dive into, the environmental toxicity of microplastics. What do we know about effects on soil ecosystems and plants growing within those soils? And there is some evidence that microplastics may adversely affect soils and plants. That's certainly the case. However, when you try to draw direct connections to something like compost application and what exactly should we expect in terms of a ecotoxicity effect, there's differences in units between the ecotox studies and the studies characterizing what exists in compost. Sometimes we're seeing effects at levels of microplastic contamination that are way higher than we would actually expect in the field. And so there's still definitely more work to do here to figure out, where is that, and if we're going to take the threshold-based approach, where is that threshold of actually observing these ecotoxic effects? How much is too much? And so just a few more comments on measurements. So obviously, if we want to monitor or regulate microplastics, we need some way to measure them. Again, no standard methods exist, and we cannot simply apply methods used for water or mineral soil to these high organic matter materials. I can talk more about what the multiple options are. There's a variety of options that range from, relatively inexpensive microscopy that is very labor intensive and is subject to some subjective bias when you're identifying the plastics visually under a scope. We do everything we can to avoid that using consistent methods with a dichotomous key, but that's always going to be imperfect. Then there's other instruments we can use to actually identify, is this a plastic polymer we're looking at? And you'd be surprised at how many natural materials look like plastic when you're underneath the microscope. So we use FTIR, and we'll talk more about that soon at UVM to try as a confirmation step. But even that becomes complicated because a lot of the packaging materials that are in these food waste streams are not necessarily simple polymers. They're like layer cakes of different things. So when we put them under our instruments, sometimes we don't get that smoking gun evidence we hope for, and it becomes a little bit more difficult to interpret. And we'll share some results to illustrate that. And then I'll jump into some of our primary research. This is a paper recently published on our study focused on the biogas potential and microplastic content of mechanically depackaged food waste. This was conducted in Vermont. And this is part of Kate's dissertation. Kate did the heavy lifting on this. I'll just quickly summarize. And so we were looking here at the mechanical depackager in Williston, trying to understand what the potential was for microplastic contamination on the back end of that machine, looking at two different feedstocks. The first is the more standard type of feedstock for these machines, which is a packaged food waste stream. So we looked at packaged ice cream pints as our packaged food waste material. And then we also looked at source-separated food waste going through the depackager. There was some interest in, what could the depackager achieve in terms of cleaning up contaminated source-separated food waste. So source-separated food waste that visibly has plastic in it, could the depackager remove that plastic with
Can you hear us now? I see a nod from Wendy.
Okay,
Okay, sorry, Eric.
we're good.
You're good.
All right.
So far, hopefully we caught you before you went far.
All right. So just to pick back up, so in this study, we're looking at a few different feedstocks going through the mechanical depackager, seeing what's the microplastic contamination on the back end. And then we also looked at an anaerobic digester that was receiving the packaged food waste as one of its feedstocks. It wasn't the majority of the feedstock. It was a proportion of it. And so we were collecting these materials, looking at the biochemical methane potential. So how much energy could we expect them to help produce using anaerobic digestion, and then looking at plastics. And so for the plastic content, we use a combination of hydrogen peroxide digestion. This helps eliminate some of the organic matter and tends to dye the rest a very white color, which can be very helpful when looking for colored microplastics in particular. And then after that process, we would sieve the materials through two sieves, one at 1 millimeter and one at 0.5 millimeters. So our lowest detection limit was 0.5. And I know Kate could tell you in detail how much more effort goes into just that drop from 1 to 0.5. It really increases the time and labor associated with the measurement. Once we had material on these sieves, we would look at them under a microscope with visual inspection, pull any suspect particles, and then we had a dichotomous key that we developed based on some previous work by other researchers, and that would help us make the determination, is this visually, plastic based on this classification system. After that, everything that passed that test would go to an ATR FTIR for analysis. Well, everything that was large enough to actually be measured by this instrument. There were some particles that were not big enough to actually be measured by this instrument. But if we could, we would measure all the representative particles of a certain class to try to say, all right, are these particles of this type actually a plastic polymer, or did we misidentify them during the visual inspection step? And so we also would weigh the particles after if possible, and if we weren't able to weigh them, we used some geometry and density-based methods to estimate mass. So we ultimately produced both types of data, particles per mass and percent weight by weight. And so just to summarize, the depackaged ice cream pints and the depackaged post-consumer food scraps had relatively high energy recovery potential. These numbers were very similar to what we would expect from these types of materials if you take packaging out of the equation. So we weren't seeing any clear evidence here that these are fundamentally different from what we would expect from ice cream or food scraps in a digester generally. And when we looked at the plastic content, the total greater than 0.5 millimeter, these are the three categories of the materials we measured in this study. You can see the depackaged pre-consumer ice cream pints had the highest particle counts, and also, or actually not quite the highest particle counts, close, but definitely the highest percent by weight plastic contamination at an average of 0.19%. And then the depackaged post-consumer food scraps had lower particle count and lower percent by weight. And then digestate, interestingly, had kind of similar particle counts to the depackaged ice cream pints, but a much lower percent by weight than the depackaged ice cream pints. So these rates, I would characterize them as relatively low on a percent weight by weight basis, and consistent with what others had reported in the literature for similar materials. Again, that's a very limited database, but these are not wildly different than what we had seen in the literature. And just as an example of why units matter, we look at these two materials, similar particle counts, 11,000 on average for one, 12,000 on average for another, but an order of magnitude difference in the mass of plastic in these materials. So that digestate kind of further along the processing stream, smaller, lighter plastics, so they add up to large counts still, but there's not as much of it on a mass basis. And then if we look at what was actually the composition of plastic within these materials, so just to kind of help give you your bearings, each column here is a unique sample, each of these stacked bar graphs, and then the top row is ice cream, the middle row is food scraps, and the bottom row is digestate. And then we have a color scheme here where the kind of yellowish green color is films, the more turquoise color is fibers, and the rose color is fragments, and the darker the color, the larger the size. And so if we look at the proportion of plastic abundance, you see a lot of that pale kind of yellowish green. And what that means is that the majority of the particles we identified were relatively small films. So these are your polyethylene films that are common in food packaging and other film materials that are relatively small in size. Now, if we look at all of this on a mass basis, you see a lot of the rows starting to show up for some of these columns. And what those are is the larger fragments. So this means when there are larger fragments, plastic fragments, in this case, not even technically microplastics, because these are greater than 5 millimeters, and most people use 5 millimeters as the cutoff. When those are present, they tend to account for a lot of the mass of the plastic. And if we kind of drill down further to show you some of the particles we isolated, we are very proud of these because it took Kate an immense amount of time to pull these out and identify these materials. But these are the most abundant on the bottom row. These are your PE films, a couple of different types of films here, including EVAC and PBAT. PBAT being that kind of seafoam green, compostable plastic.
What's EVAC?
What?
What is EVAC?
EVAC's also
ethylene vinyl something. It's something. So a lot of these films, we used a shorthand PE film, but based on the FTIR results, we think that many of them are actually a whole bunch of different types of plastic polymers, and also like adhesives and pigments, all kind of found together because different types of plastics have different barrier properties. Some are really good at, keeping moisture out. Some are good at keeping gases out. So the food packaging industry will laminate a whole bunch of them together. So it's great for, food packaging properties. It's really hard to identify because it's not one thing. It's like many, many different things. So for this project, we grouped a lot of those together as PE containing film.
I was wondering if you have the ability to ask for packaging specs from the industries that are supplying the pre-consumer waste. Those exist that tell you exactly what those layers are. I don't know if, but it depends on.
I think if you had a single origin food
Mm-hmm.
waste stream, you could potentially do that. But with anything that's post-consumer, who knows where all the different stuff is coming from.
And we took steps to try to isolate these feedstocks in our study, but we were seeing some evidence that with the deep packager, you would see some material coming out in like the ice cream or in the digestate, which is the ice cream then put through an anaerobic digester, that are not part of the ice cream packaging. So there's some cross-contamination that can occur. And then the greatest mass, again, when you have fragments, so either large films, or large films. So you can have large films that are pretty big, or you can have different types of fragments like PET, PLA, EPS. So to jump into the compost study, this is the second study. We learned a lot of important kind of lessons on methods in that first study, and then we applied them in the next study on compost. Again, we looked at 20 composts across Vermont, the majority of which had food waste as a substantial feedstock, but also some where food waste was not a substantial feedstock. This is the first study of this kind in Vermont, so the questions were relatively simple. Are Vermont composts contaminated with microplastics? Do we see any correlation between the number of microplastics and the overall mass of plastic contamination, and what type of polymers are most common? And so for these are just all 20 samples. On the left in the purple is the count. On the right in the blue is the mass numbers. We actually developed a system for compost using different confidence levels because compost was even more problematic in terms of having a lot of plastic look-alike natural materials. So we are most confident in the dark bars here. The light portion of the bars are suspects, but ones that did not meet enough criteria for us to have a larger amount of confidence in. So if you look at the numbers, they vary a lot from non-detects. We did have a few non-detects, even when you consider only the higher confidence particles. And we also see like the highest amounts kind of, around that 1,000 particles per dry kilogram, which is kind of a number we've seen in the literature, this range from really low numbers into kind of the 4 digits. That is what we saw for counts. If we look at mass, that number, the highest number of our more confident particles is right around 0.05% weight by weight. So for those of you who are involved in any way in kind of the debate about potential policy on this issue, it was 0.5% weight by weight that was being considered, with film plastics being 20% of that, meaning that the limit on film plastics back when there was a proposal on the table for monitoring and regulation would have been around 0.1% weight per weight. So even if these were all film plastics, all of these materials would have been under that number based on our estimates.
I have a question.
Yeah.
About the food waste stream. Was there consistent, like, across 20 different composters, did they have, I mean, it's food waste, but I feel like there's still different varieties of waste streams that we call food waste. So I'm curious, do you have any?
We collected as much information on the feedstocks as we could, and we will have that as supplementary information. We're working on getting that paper published soon.
Okay.
I don't have that today, but there was variability. There's variabilities in the proportion of the feedstock. So that's all of that. This is kind of not a super controlled study. This is more of a survey what's out there.
Okay, thanks.
And we were able to kind of break it down by the higher food waste feedstock compost and the lower or no. And I think our cutoff to be in the lower or no was less than 5% of the feedstock. And this is preliminary data, I would still call it. I mean, I feel good about these data, but this is not necessarily proof, but it does provide some evidence leaning towards that food waste is a key source here. We tend to see higher numbers when food waste is involved, which makes sense. And just that number for the higher food waste compost of 0.017% weight by weight is really similar to the number we had for the digestate we analyzed. So they were really, really close, actually, those two numbers.
What were the feedstocks for the no food waste?
So these are things like horse manure in some cases, more manure-based feedstocks or other agricultural residues, not necessarily anything coming from food waste streams.
But still with microplastics.
Yeah. And so that food waste, we think, is the dominant source within these materials, and packaging is ultimately really the source within the food waste stream. But, we have other things other pathways exist. There's litter, there's atmospheric kind of movement of microplastics through the air that can enter these systems. There's plastic used on site for various reasons. So even, we did get non-detects, but in the majority of samples, we still found something, even if it was a very small number of particles or a very small mass of particles. So just some key high-level takeaways. It's a systemic challenge. Just want to stress that point. I think, whatever from a regulatory or policy perspective, this is something that kind of deserves broad focus. But we still are trying to understand, like I just mentioned, there's other pathways of microplastics to the environment. These types of food waste derived materials are one pathway, but we don't necessarily have good information on how that pathway compares to something like use of plastic mulching on farms. If you were to try to press me on which is a bigger source of plastic, I wouldn't know what to say right now. We still have a lot of work to do to try to understand these different flows. So again, we think food packaging is the dominant source within these compost and digestate that we've looked at so far. And then I'll turn it over to Kate to quickly go over some of the ongoing work right now on life cycle assessment.
Yeah, so I'm Kate Porterfield. I'm a PhD candidate at UVM. Been doing a lot of work with microplastics in food waste for my dissertation. I'm really excited about this last chapter that we're working on right now. It's been in the works for a long time, and it's kind of arisen from all of this previous work. So we're taking a lot of lessons learned from the past 3 years and then trying to apply it to this newest project. So for those who might not be as familiar with life cycle assessment, it's just kind of like a very broad description for modeling approaches that try to measure the impact of a product or process over its entire life cycle from cradle to grave. So the thing that you're measuring can be really different. Often it's CO2 emissions or CO2 equivalents. So when we talk about like something's carbon footprint, that comes from a life cycle assessment to calculate the carbon footprint of that product. So we're trying to address 3 big questions with this new project. The first is what environmental benefits and burdens are associated with different food waste management strategies. And we're primarily focusing on landfilling, kind of the status quo in a lot of places, and then composting and anaerobic digestion. Obviously, there's some other strategies, diverting food that is still edible to food shelves is a great option when possible, but a certain fraction of our food waste is inevitable, and for that fraction, a lot of it's going to composting or anaerobic digestion. And then one novel aspect that we want to add to this LCA is try to calculate a flow of microplastics to agricultural soils under these different scenarios. And then finally, we want to know, do the characteristics of the food waste stream, so how much packaging is there in the first place, the total solids content, the phosphorus content, the nitrogen content, do those characteristics influence what would be the optimal strategy to manage that food waste stream? So to answer these questions, like I said, we're conducting an LCA. So the goal broadly is to inform food waste management decisions under the new diversion mandates established by Act 148. A functional unit in LCA speak is basically like, what are you calculating the impacts of? So in this case, we're trying to calculate the environmental impacts of managing a ton of mixed post-consumer food waste, including all the contaminants that are in it. So that's a big distinction from a lot of the food waste LCAs that have preceded us. Often they won't consider that the food waste stream is contaminated at all, which I think we're coming to realize is usually not the case, or they'll assume that whatever separation strategy they're using is perfect and that it just perfectly removes all contaminants. I think we also know now that that's not usually going to be the case. And the scenarios we're looking at, like I mentioned, landfilling, anaerobic digestion, and composting, and the impact categories that we're focusing on. There's tons of different impact categories out there. You'll hear like human cancer impact, human non-cancer impact, acidification, all sorts of ones like that. But we're kind of focusing on the ones that we think are really applicable to this particular issue, and that kind of fit with our expertise as nutrient cycling people. So looking at global warming potential, and that's going to be units of CO2 equivalents. That's probably the one people will be most familiar with. And then eutrophication potential. So we do a lot of stuff with water quality, very interested to know how different management strategies are going to affect water quality. And so for that, we're looking at both marine eutrophication potential and freshwater eutrophication potential. And a very simplistic shorthand assumption that people use for this sometimes is that marine eutrophication is driven by nitrogen and freshwater eutrophication is driven by phosphorus. Not always the case, but it's a good proxy to help us try and come up with a metric for each of these things. And then this new category we're introducing is plastic pollution. So we're still working exactly on, what the units of that are going to be. Right now, in the current iteration of the model, it's kilograms of plastic. But we're hoping to introduce, more of a kilograms plastic equivalent thing that will take into account which fraction of the plastic is biodegradable and which is not. So we'll have both like a mass and a duration in the environment dimension to it. that's in the works. So don't be overwhelmed by this colorful diagram I just put up. This was actually really helpful for me to make this morning to start to think through how I want to present this model when we publish on it, hopefully soon. But I'm going to walk through it real quick. So starting with the purple box, this is going to be our inputs to the model. So this is going to be whatever organic waste stream we're modeling. I'm starting with source separated organic waste because there's the most information out there about it. And so what's unique about this model is that we're not just thinking about a generic source separated organic waste. For this model, you can actually change the characteristics of it. So we're going to input, the total solids composition, total volatile solids, how much plastic contamination is starting in the waste stream, carbon and nitrogen and phosphorus content. And then those characteristics of the food waste stream are going to influence what happens in the model, and it's going to influence the outputs. So this is just an example of the very simple depiction of the anaerobic digestion scenario, but we have a model for composting and one for landfilling as well that I'm not going to show today. But basically, it's just a very simple kind of mass balance approach. The food waste goes into the mechanical packager, and some portion of the plastics are rejected and will be sent to a landfill. Some portion sneaks through and goes to the organic fraction, which goes to the anaerobic digester. And there, the organic material is converted to biogas, methane, and that can be used to generate electricity. And so in the model, it's credited, generating electricity is a credit. It offsets electricity generation elsewhere in the grid. We're using the Northeast electric mix as our offset for the model right now. It also will emit some fugitive methane emissions, so that's going to be a knock to the global warming potential. And then the digestate that's left over is going to be land applied. And so in that component of the model, we calculate, how much mineral fertilizer does not need to be produced because of this digestate that's being applied, how much phosphorus does not need to be mineral phosphorus does not need to be used because of this. We estimate how much plastic is sent to the soil in that digestate, and then also how much nitrogen and phosphorus is not taken up by plants and is kind of out there active in the environment and could potentially contribute to eutrophication. So those are the foreground emissions that we're modeling directly based on characteristics of the feedstock input. And then there's all the background emissions that we can't, we know are existing, but we might not necessarily be able to model really directly. But we do have this really great database where we can kind of connect our foreground model into this bigger background model so that when I credit this process with offsetting nitrogen fertilizer elsewhere in the model, there are emissions being subtracted for nitrogen fertilizer that is not needing to be produced somewhere. So this is a very simple view. It looks like a crazy amount of arrows when we actually put it together in the software we're using, which is called OpenLCA. And then the outputs that come out of this are that the global warming potential, the marine eutrophication potential, freshwater eutrophication potential, and plastic pollution potential. So the idea is we would run, the same organic waste through the anaerobic digestion model, the composting one, and the landfilling one, and then we could compare the impact across all four categories across those three scenarios and start to see if there's a clear winner or if there are just trade-offs and we need to decide what's more important, global warming potential or some plastics going to the soil.
So this model, at risk of sounding really dumb right now.
No.
This model is showing, so you're looking at like the three things, whether it goes into the landfill, whether it's
composted
or digested. So this is showing.
This is just anaerobic digestion.
Your
So this is
scenario.
just how I was get the numbers for anaerobic digestion. I could do 2 more slides and there would be something similar to
For
this.
each scenario.
Exactly, for composting and landfilling. But I don't think we need to go through that again.
No, got it, I'm with you.
So this modeling approach is novel from other LCA studies of food waste because we're really focused on nutrient mass balance. So what goes into the model is going to affect the impacts that come out of the model. And you can change the characteristics that go in. So we want to build something that's, you know, going to enable us to compare multiple different scenarios. And then the other, I think, really novel aspect of it is trying to include microplastics as an impact that has not really been done effectively yet, although there are a lot of people trying to figure out how to build microplastics into LCA studies. And then the other thing about it that I'm really excited about is you don't have to just input static numbers for each of the parameters. So if you know that your feedstock is like 1 plus or minus 0.5% nitrogen, you can put 1 plus or minus 0.5% into the model, and then it will output results with a mean and standard deviation. So we know that like we can never really pinpoint reality with these things, and so we wanted to build something that's going to give us a distribution of possible answers, and then, you know, we can see where the best case scenario is and where the worst case scenario is, and not just rely on means all the time.
Great, thanks, Gary. So that's all we had. It took us a little longer. Hopefully we still have some time for questions, but we'd love to hear, you know, what are the concerns that have been expressed so far about microplastics? What's going on with some of the state's current efforts? And yeah, or just answer any clarifying questions about the presentation.
Go ahead, Amanda, do you have yeah, go ahead, Amanda.
Hi, yeah, so we have an anaerobic digester we put in 2006, and we're currently going under a redevelopment of it, and instead of making electricity, we're going to be using gas. We're going to be producing gas. So one of the biggest hurdles that we've had in the regulation is, are we taking substrates? Are we taking any materials other than manure? So there's a really strong push about doing any of that. And so I guess I recognize our purpose here on the AIB is to showing potential and to help, you know, navigate some of the regulatory work around this issue. But I guess, what have you guys been hearing? And I'm just like a little curious. This is just a total about face for the feeling we have on anaerobic digesters and what we can put in them and what we can put on land and land apply it. I think it's very interesting, and I definitely want research to continue because I think it really could change things. But I also, in the world that we're dealing with today, I'm not sure how popular this would be.
Yeah, I will say part of our current EPA-funded study included interviews, and I'm not sure if we got up to your farm or not, but we interviewed farmer digester operators. This question about kind of food waste materials going on to into farm digesters is kind of the question you're referring to, and we interviewed several different farmers to understand their perspectives on that, and we'll be sharing that data probably within the next year as well. And I can't say too much right now in terms of kind of generalizing, but I can say that, you know, there's a trade-off here, right? This is a way to boost methane production if that's a goal for the digester, but there are concerns out there about the potential contamination risk. So I think figuring out ways to reduce that risk is really critical right now. And I think with more kind of efforts upstream to, you know, focus on reducing the amount of packaging to begin with, focus on moving towards better materials over time, focus on trying to enhance the quality of source separation by people, or maybe continue to innovate in the mechanical depackaging space. Hopefully, all of these things combined could help reduce that risk. But right now, we do see it as a bit of a trade-off where you have this energy potential there. It's real, and it could be very important for certain digesters. But, you know, right now, there is going to be some level, even if it's low, of microplastic contamination risk.
So have you evaluated different methods for reducing the microplastics in depackaging? Is there?
No, but that's something that definitely should be studied. I mean, this is, these machines, you know, to our knowledge, there's a lot of choices in terms of exactly how you run the machine, exactly, you know, how much water do you add, at what speed do you process things. There are ways, I think, that we could better understand, you know, how to optimize removal for different food streams. I mean, I will say we just looked at one depackager, and there are multiple different models out there, so I wouldn't pretend to generalize that our findings represent every depackager out there. But, you know, the way I look at that data is there's definitely some getting through, but the manufacturers have said 99% removal, and, you know, based on all the kind of back calculating we've done, that it seems like they're, you know, above 99% removal, most likely, and yeah, which to me is still impressive, but we'd love to get even lower, right, in terms of what comes out the back end eventually.
Are there any standards for the amount that?
I think right now there's a working group, well, Morgan, you might be able to update me more on this in terms of, there was the stakeholder working group on the depackager. They released a report very recently that had some recommendations, but I'm not sure what the next step is with that.
I don't know either. Stephanie,
Yeah,
do you know?
Steve Cash was the agency's representative on the
Right.
stakeholder group, but I'm not sure what the next steps are either.
I can find that report for AIB members.
Yeah.
I can take that as an action item so that we can share that. Yeah, I think the report, I'm not sure if they're even meeting
I don't think they're still meeting, yeah.
I think it was done with the
after the
report.
report.
Yeah.
Mhm.
Yeah.
What was that task force?
It's
It was
the
a depackager
packager stakeholder.
stakeholder group, and it was run by GC.
Yeah.
So Steve Cash was our representative.
Have you ever looked at microplastics and biosolids?
We haven't personally, but there are several papers out there,
A
and
large body of literature
Yeah,
documenting
they-
microplastics.
they're there.
Yeah, they're
for
there.
sure.
I mean, how do they compare to the depackaging? You know?
There's a lot of fibers in biosolids because polyester clothing, yeah, and washing machines. So gets to the units question again. Probably really, really high numbers, but then the mass, you know, varies probably.
How about, you had mentioned earlier on about ecotoxic or toxicological effects or ecotoxic. What can you say about that?
So there's been studies looking at, you know, individual soil organisms like earthworms, for example. There's been studies trying to understand, like, do microplastics affect some key soil functions that are carried out by microbes? And I think the answer kind of, it's unsatisfying, but it's a big, it depends. You know, some of these studies find effects for certain plastic types, but not others, certain plastic sizes, but not others, certain soil pH conditions, but not others. So in general, I think we can say, yes, there are potentials for toxic effects, but in terms of knowing, you know, exactly how to go about defining like a regulatory threshold for like what level of microplastics is likely to elicit that effect in the field, that's where there's still work to be done, I think.
Yeah, I was thinking, you know, when a digester operates, and then it gets land applied, like there's one application, there's another application, there's another application, so there's the stacking or cumulative effect of digestate on land.
Yeah.
So how is that incorporated, or is that incorporated in the study that you're?
Well, that was our big motivation for
Okay.
making the mass balance-based model, because all of our previous work was, I think of it as like a snapshot in time. Like, you're looking at like, what is like basically the concentration of plastics in this material. But like you said, like you know, if you're applying it, a whole bunch, like a lot of it over time, it's gonna accumulate in the soil. Which is why the biosolids regulation, you know, has like concentration limits, but then they also have cumulative loading limits on like heavy metals and stuff.
Yeah.
So in one of our papers, you know, we suggested like, if you eventually were to be regulating microplastics, like it might make sense to have a similar model. But yeah, that's why we're making a mass balance-based model, because then you can say, you know, if you're starting with this amount of plastic in this amount of food waste, and you pass it through these different management pathways, like per unit, how much plastic are you going to be putting in the soil? And it's all going to come down to how much plastic are you starting with, and then how effectively are you separating it out before it makes it to land application.
But yeah, I would just reiterate that point, that I do think it's important to think about cumulative loading eventually. These are particles, most of which will not break down, and break down very, very, very slowly. And so that cumulative piece seems important, more in line with heavy metals that tend to stay put in soils. Versus, I know you focus on other contaminants that are kind of short-lived, but potentially highly toxic. This is a very different situation where they're long-lived, maybe lower toxicity, but can build up over time.
One sort of, and I know because we're talking about a million different plastics, is there a general range of persistence?
In the environment?
Yeah.
Well, some of the compostable plastics should break down quickly. But then like hundreds of years for some of the other ones, it's totally going to depend, like how big is the piece when it starts, and like is it exposed to the sun, and
then is it really gone, or are we just breaking it down into smaller and smaller pieces? But you know, I do think ultimately, if we really want to kind of seek the best solution to this problem, it is upstream in packaging design. And this packaging was not designed with the kind of circular economy as part of the model. It was designed to serve food safety purposes and advertising purposes and go to a landfill. So, you know, I'm hopeful that over time we have better materials that present less risk. I think it will always be hard to get every little fragment of packaging out of the food waste stream, but if the ones that are left are more benign than they are today, that would be a big win.
I know that we're close on time, but I wanted to see if Sarah, Owen, if you had any comments.
Not really. I mean, I did present at that depackager work group that was just discussed. I said November, but I meant November last year, 2022. I don't know where a year went. Similar to the answer of, you know, are they ecotoxic? Looking at the information that was published as of 2022, there's certainly some information that suggests they're toxic to humans as well. You know, inhalation is an obvious pathway of concern. But even with ingestion, which is more relevant to these conversations of food packaging waste, you know, a similar answer of yes, sometimes they're toxic. It depends on the particle size, what's been added to the particle, any other chemical constituents of the particle. Although a paper was just published in this month in Journal of American Medical Association that looked at plaques. So somehow the people's plaques got pulled out of their arteries and they were dissected for microplastics. And the people who had microplastics were, I think, I don't know, twice as likely. I don't remember, I'm not an epidemiologist, but much more likely to have had some sort of adverse cardiac event. So general consensus is yes, they can be toxic to human health, but, you know, similar to what everyone's saying, what do we do about it? You can't undo, you know, decades worth of plastic that's been put into our environment. So. That's all.
Thank you.
Sorry, I'm always like, yeah, I'm like, God, why do I even say things anymore?
Makes us think, Sarah, makes us think.
Fun to watch somebody else try it.
And on the inhalation piece, we've seen some things to suggest that like just being in this room, breathing is like, might be the worst exposure that we all get. Microplastics, because we have all these carpets and clothes that are all made with polyester, you know, and like airborne. I mean, when you see the dust, you know, at a certain time in the afternoon, those particles suspended, like a lot of those are microplastics that we're breathing in to our lungs. They're kind of everywhere.
I travel with a little air purifier right next to me.
I have a really intense air purifier now after doing it.
I don't think that the membrane is small enough to take out the particles that are going to affect your lungs. Sorry. I think microplastics come in such tiny, I mean, you're talking about 0.5 millimeters, I think, in the presentation, but
Yeah.
I mean, we're talking like nanometers are
Yeah.
present and affect small enough to get into every part of our body and affect public health. So, yeah, sorry, ruined your air filter idea. It'll help with the big ones.
I think for regulatory agencies, a key question is with I see some parallels with PFAS or something that's widespread. It starts to become, where are we looking for it? Because where we look, we probably get a detection. So I think programs need to be established to try to really isolate what are the key pathways of exposure, and do that thoughtfully so we don't just kind of play whack-a-mole where we look here, oh, there's microplastics here, we look, there's microplastics there. I think that would be a more reactive approach that could cause confusion.
Morgan, I know we're over time, but I think I heard you say we're searching for new topics to work on. I would ask that we not work on this one.
I will write that down. Do you wanna give a reason? Do you wanna?
Well, I think if we're going to work on it, we would have to identify some tangible goal, right? So, for example, removing microplastics from our food waste source is not a achievable goal. Writing a report, sure. Reports already exist. So I think if we're going to take it up, we should make sure that whatever we're going to do, we come out with something usable, not just reaffirming what we all know.
Thank you. Sorry, I wanted to write that down. Yeah, that was so good, Sarah. Well, thank you, and thank you, Eric and Kate, for coming down. Are there any other questions before I ask for public comment? Okay.
I'm
Go ahead,
sorry.
Steve.
I do think we ought to keep in touch with your research. So, you know, I mean, I agree with Sarah that we don't just want to take it on, you know, without having a goal in mind, but I do think that it's good information to be aware of, and potentially there could be some role for the AIB in making a recommendation regarding some of the materials that are used in agriculture. So I think it's just being aware of what you're finding and where you're going, I think it'd be useful. So maybe just sort of put that as something to keep out there, you know, for a recurring topic, you know, at some point.
As Sarah mentioned, sometimes the concern is the chemical additives, actually. So in that case, you just might want to keep in mind, if you're thinking about different chemicals, are they potential additives in the plastic packaging that might be another source that they could get into the environment.
What's your next, in terms of your research program, is there another logical report time or endpoint that would be worth coming back to tell us about?
I think the redundant study that Kate mentioned, I think it would give us a better sense of the scope and scale of this potential issue.
Okay.
In a new way.
Right.
Yeah.
Okay.
So, and You're graduating this summer. Sometime in the fall.
Okay, well, that'd be appropriate. So we'll keep that in mind, and you know how to get a hold of us, and we know how to get a hold of you. That's good. Yeah, we really appreciate you coming.
Yeah.
Yeah.
Thank you.
Thank you. Okay, we have a few, members of the public, so I just wanted to open it up. Is there any public comment at this time? Okay,
And reminder when the next meeting is, maybe?
yep, so, your calendar should be up to date, but let me know if not. Next meeting is May 20th. Monday, still 1 to 4. So thank you all, and enjoy the rest of your Monday.
Thank you.
Thanks.
Thanks, guys.
Thanks for coming.